Hey Cam followers, welcome to another one of our lives. It's me, Esme here, and I'm joined by the lovely Andrea Tarr. Andrea, I'm going to pass straight over to you. Do you mind explaining to any followers of ours who haven't watched one of your previous lives who you are and what you do?
Of course, yes. Thanks. Hello. Hi, everyone. And thanks, Esme, for inviting me to come onto this. My name's Andrea Tarr. I'm a pharmacist. I've been a pharmacist for about just over 35 years now. So I've got very sort of pretty broad knowledge and experience of medicines regulations. I've worked in the NHS, in the pharmaceutical industry. But for most of my career, I was very fortunate to work on a very highly respected independent medical publication called the Drug and Therapeutics Bulletin, with some real pioneers in independent medicines information. And a lot of what we were doing was, what we were mainly doing was scrutinizing evidence on medicines, a lot of which involved examining randomized controlled trials. Since then, I've founded a much needed independent resource on veterinary medicines called Veterinary Prescriber. It's an online CPD and reference resource. And again, I've continued to do the same thing, which is to scrutinize evidence on veterinary medicines. So a lot of what I do from day to day is read randomized controlled trials.
Yes, and that's what we're going to be talking about today. If you've not heard of Veterinary Prescriber before, I can highly vouch for it being a really, really useful resource. I've used it myself a number of times since meeting Andrea, just to learn about different medicines and what's going on. But we'll put a link up to that at the end so that anyone who wants to go and have a look can do. Do you want to talk to us a bit about randomized controlled trials then?
Yeah. Well, what I'd like to do first is just to kind of tell you a little story about my garden, actually. You might think it's odd, but it'll make sense at the end. So this house, I moved into this house about 1.5 years ago. It's got a sort of a strip of lawn at the back. And when I moved in, it had this kind of mossy area right in the middle, as a bit of unsightly. And I quite like gardening, and I wanted to fix that, make the lawn look better. So I had the idea that the problem might be due to poor drainage. So what I did was to kind of go out several times with my garden fork and make some drainage holes in the sort of mossy patch. I did that several times, and then with the aim of improving the drainage. So I did that, forgot about it. And, we've had, over the last summer, the summer that's just gone has been really nice. I've been spending quite a bit of time in the garden, and I've really forgotten all about this, but, I was outside a couple of weeks ago enjoying the last of the nice weather with some other members of my family. And it struck me how lovely the lawn was looking, and this sort of mossy area had disappeared, and the moss had gone, was replaced with lovely green lush grass. So I proudly said to the others, look at the lawn now. That's due to my hard work. I've been out there digging. It's obviously fixed the problem. And then my son said, yeah, but wait a minute, remember that tree on the other side of the fence? It used to have long branches that kind of spread out over the grass and stop the sun from, shaded that area and stopped the sun from getting to the grass. Do you think it's more likely that it was lack of sun that caused that unsightly patch? So, well, I had to admit that was, a possible reason why the grass is growing now. But, I felt very proud, I had a real sort of investment in the sort of hard work I put in, and I felt a bit biased towards the theory that it was the lack of drainage. But really, it could have been either of those two things, or it might even have been just, the particular weather conditions that we've had over the last year or so. Mm-hmm. So, and the point I'm really trying to make is that, I sort of made an intervention to solve a problem, but when we make an intervention, it can be really difficult to know exactly precisely whether the intervention you've made has really made that difference, because there can be all sorts of other things going on, things that you might be aware of or things that you're completely unaware of. And it's very difficult to sort of pick it all apart and see what's actually having an effect.
Yeah.
And it's the same with any kind of health intervention, because often there are lots of things, different things going on. I mean, you know, I'm sure we all know we've had a personal experience of kind of making some sort of change. You know, you might feel you decide you're going to do more exercise, change your diet, drink less. You might, and often we start doing lots of different things all at once, and then, you know, we see a change. You know, perhaps we lose weight, but it's difficult to attribute the weight loss to, you know, any particular intervention. So, and with health interventions, there can be lots of things that are going on that you might not think about. I mean, with some diseases, they're self-limiting. They just get better by themselves. So, for example, you might have had a cut, and, you know, some people like to put some antiseptic cream on the cut, and the cut gets better. And, you know, you might believe that the antiseptic cream has helped make the cut better, but actually the cut would have got better by itself anyway. So, same with some, you know, infections like eye infections, that kind of thing. Or if we're talking about a long-term illness, you know, many illnesses kind of go up and down. You know, things like epilepsy comes and goes, and osteoarthritis is a really good example. It's a disease where the symptoms kind of come and go. You get flare-ups and so on. So that's why it's difficult if you make if you try some sort of treatment at any time. You know, it might be that it just happens to coincide with when the disease was, you know, coming to the end of a flare-up and was going to be getting better anyway.
Yeah.
So that's why we need to be, you know, we need to find a way of testing if an intervention is really working. And the reason why it's important to do that is because, you know, with any health intervention really, there's usually going to be some sort of a cost. So there might be a financial cost, but also there could be a cost in terms of some sort of harm. So that's why it's so important. You know, that's why we can't really rely on personal opinions about whether treatments are working or not, because there are all these other things that might be going on at the same time. And the only way to get a really objective idea of whether a treatment is having an effect is to find a way of ruling out all these other sort of confounding things and biases that could get in the way of knowing whether what you're doing is actually having a real benefit.
Yeah, I think that's even more important in animals, isn't it, with our pets, because we introduce a bias as well. We want the treatment to be working because we do want our dogs to be more comfortable, be moving better, and so we have to know that whatever intervention we're giving has good evidence, so we're giving the best chance for that to be limited. So what the outcome you're seeing is truly there, not what you want to see. If that makes clear sense what I said. So I think having that evidence is really important, so that we can say, actually, let's try this drug, and we know that it's had this result in the study, but we also know that it has had these side effects, so we know what to look for. And I think we've talked about this before, but we talked about evidence-based medicine. That's something we're so passionate about on CAM, making sure that we're giving the best evidence we've got. And that's what you do, a vet prescriber, isn't it? Talk about evidence.
Yeah, exactly. Because some people might say, look, well, you know, there's this sort of very believable theoretical reason why you should use a particular drug or something, because we know it interacts with this particular receptor, or it does this and that and the other. But that isn't enough on its own. You have to know that it is having an overall benefit on, you know, in the animal, because I mean there are plenty of examples in human medicine actually where things have. You know, they've been used because it seems like a good idea, but actually when they've been tested in randomized controlled trials, it's found that they're not having the benefit that you thought they might be having, or they're causing more harm, and the harm is outweighing the benefit. So it is really important to sort of unpick all this and know exactly what you're using is having more of a benefit than a harm or a cost.
Yeah, absolutely. So how do we find out if a treatment works? What do we do?
Well, you have to kind of do it in a way that cuts out as much as possible all these other things, and that is the way you do that is with a randomized controlled trial. So the fundamental parts, the fundamental features of a randomized controlled trial are that you have a control group, first of all. So say you wanted to find out if a particular new drug reduces pain in a dog with osteoarthritis, then you could give it to a group of dogs with osteoarthritis. But if you give all the dogs the same drug, you're not going to find out anything very useful, because it could be you're not cancelling out the effect of other factors that might be sort of muddying the water. So in a randomized controlled trial, you have usually two groups of people or animals. You have the test group. They're the group that are receiving the drug or intervention that you want to test. And there's a control group, and they should be undergoing exactly the same experience as the dogs in the test group, and everything should be the same apart from not receiving the active drug. So the two groups should be as similar as possible, and they should be treated in exactly the same way. And the way to make sure that the two groups are as similar as possible is to randomly allocate each dog that's being entered into the trial to one or other group. Mm-hmm. So that means that there's no sort of either conscious or unconscious action, which means that a dog is more likely to go into one group than another. What I mean by that is if there was no randomization, the researcher might be, for every dog that came along that was going to go into the trial, the researcher might think, oh, well, actually this dog is, got more severe arthritis than the other one. So, well, it'd be better if using the one in the group receiving the test treatment. So randomization aims to kind of rule out that kind of bias. So in a randomized controlled trial, then you've got usually two groups, and the two groups will be, should be very similar in terms of all the various sort of features like weight, age, breed, disease severity, all those kind of things that could bias the result at the end of the day. So if there's a sort of an imbalance between the two groups, then that might have an effect on the result, which may make it a sort of an untrue result, if you see what I mean. Have I explained that well enough? Yeah. And there's a further important feature, and that's I'm sure people often have heard that as well as the term randomized controlled trial, such trials are usually described as being blinded, either single blind or double blind. And the reason for that, blinding, or another name for blinding, is masking. And the reason for that is that when the trial is ongoing, well, from start to finish, anybody who's involved in sort of making measurements in the trial, in assessing the effects of the treatment, should be completely unaware of whether the animals in the trial have been assigned to the test treatment or the placebo or the control treatment. That's because if they were aware of it, then that can also introduce bias, because if you think a dog is having the real treatment and isn't in the control group, then you might expect to see better results, and that might influence the way you recorded the results. So that can also bias the end result.
Yeah, absolutely. So we've got a couple of questions as we go, if that's okay. So Julie said, why do you think there's often a very small group of dogs in some trials compared to some human trials?
Yes, no, that is a good question, and it's true as well. I mean, I haven't come on to that yet because talking about sort of fundamental things, the randomization, the control, the blinding, and so beyond that, there are other really important features that affect the outcome of a trial. I mean, perhaps I should say, first of all, when we're looking at randomized controlled trials, what we're trying to do is determine how confident we are that the result is a true one. How confident could we be the result is reliable and true. And part of that is the sample size of the trial. Because so when I read a randomized controlled trial, one of the things I look at is the explanation about how the trial was done, and in particular the statistics section. Now, I'm not a statistician. I know very little about statistics, but I do know that the researcher should explain to me why the trial has got the number of dogs in it that it has. And so when researchers are designing a trial, they should be doing a calculation to find, well, first of all, they should be deciding what it is the trial is going to find out. And usually they should be just one or two very important questions that the trial is trying to answer. And so once that, and usually there's one important question, and that's called the primary outcome. Because the more outcomes you've got, sometimes trials are looking at lots of different things, and that is very kind of sort of worrying in some ways, because the more things you're looking at in a trial, the higher is the chance, the bigger is the chance that, the bigger is the possibility that you're going to get a positive result that's just due to chance. So by focusing on one question to answer, that means that the trial should be designed around that question, and the researchers should be calculating, they should be working out what sort of change would we expect to see, and that change would be not what change we expect to see, what change do we want to see, what size change would be important to see. That's what researchers should be doing, because that's what we want to know. We want to know that dogs are going to be considerably more pain-free, not just a tiny, tiny bit more pain-free, but clinically importantly more pain-free. So the researchers should work out what difference they want to find in the trial, and work out how many dogs would need to be entered into that trial in order to get a result that you could be confident was a fairly sort of true result.
Yeah.
And unfortunately, a lot of the trials in animals are very small. Yes, they often involve only sort of 20 or so. And yeah, they're probably in that case too small really to show results that are reliable. And that is a problem. There are a lot of very poorly designed and reported trials that are published. And you can compare that with, I mean, it's not true for all animal trials. I mean, in this country we have good medicines regulation, and so if in order to market a veterinary medicine, a company needs to prove efficacy, safety, and quality. And in order to prove efficacy, what they have to do is do randomized controlled trials to show the effect of their drug. And so, for example, a drug that we reviewed very recently in the veterinary prescriber was Rapiprant or Galiprant. There's one main big trial on that drug, and that was, that was a good size. That was in nearly 300 dogs, which shows, that's the kind of size you need to be able to show
trust
a reliable result.
Yeah, because I think obviously there's ethics involved, so I imagine that can be a stopper on some of the trials depending on why they're doing them. But as you say, we need to have a big enough population that we can trust in what they're telling us.
Mm-hmm.
I just from a personal experience, we had a drugs rep come to my work and talk to us about a particular product, and they were recommending it over another product that we already had in the practice. And they said, oh, this product's 100% accurate and it's so much better than the current product you're using. And then when you looked at their trial, it was on 10 dogs. And I said, well, how can you compare the two studies where the other one had hundreds of dogs? And yeah, they'd had the less percentage of efficacy. So they would say 98% rather than 100%, but you can't compare hundreds of dogs versus 10 dogs. It's the population's so small, you can't trust in it. But the problem is they were giving out the stats, but not talking about the population size. So that's where it gets quite confusing for people who aren't comfortable looking at the papers. It can be quite a minefield knowing what you're looking at, can't it?
Oh, definitely, yeah. I mean, it's really not easy because, unfortunately, like I said before, there are a lot of trials that are not very well designed and not very well reported. And, and it's not enough just to have the description in the title that it's a randomized controlled trial. So what we have to do, when we're looking at trials is we're looking at how good a job has the researcher done in ruling out the bias and trying to produce a reliable result. We're looking at all of those things, including the sample size. And another thing about sample sizes, I mean, all trials are, it's a trial in a sample. So if you repeated that trial in another sample, you might get a slightly different result. And if you repeat it over and over again, you'll get slightly different results every time. And there's always a play of chance as well. And so, and it might be that the trial that you're seeing in 20 dogs arose, that result arose through chance alone, perhaps. You don't know. So you can't rely on results of one or two very small trials. And that's what people have heard of systematic reviews. That's what they try and do, is to put the results of many trials together so that you've got a sort of a bigger overall sample to try and get a more accurate estimate of the real effect of the treatment.
And Lynn's just asking about how, so she said it must be quite difficult to control all the variables like weight and lifestyle. So how do researchers try and do that? How do they try and limit the variables?
Well, first of all, by defining the type of animals that go into the studies. So often it'll say, we're only going to take in animals that are between certain ages, perhaps, or that are between certain weights, maybe, that have this particular severity of disease. Often they'll rule out animals that have any sort of concurrent illnesses, like renal disease or kidney disease or liver disease. So, that's one way of kind of sort of narrowing down the kind of type of animal that goes in. It's the same in human trials as well. And there's a sort of a problem with that as well, because if the trials are only done in animals that are otherwise healthy and they've got osteoarthritis, but they haven't got heart disease and so on, then that's not really a very good reflection of sort of real life drug treatment, where vets often might have to treat a dog that's got osteoarthritis and some other diseases as well. So it's a problem with sort of transferring the information from the randomised controlled trial scenario to a real life scenario. So that's one way, but the other way then is randomisation, to try and make sure that the two groups are sort of evenly balanced in terms of the various factors and so on.
Okay. So would you like to go through the fundamentals of a randomised controlled trial? So we've kind of summarised a few of them, haven't we?
Yeah, well, the fundamentals are having a control group, that the animals are randomised, and the other thing is masking. So if you're reading a published randomised controlled trial, I mean, you don't take it as read that all these things have been done just because it says in the title it's a randomised controlled trial. One of the things you're doing is checking that these things have actually been done to a kind of high standard. So when a randomised controlled trial is published in a journal, it's usually published in a kind of a standard format. So there'll be first of all, there'll be an abstract, which is a sort of summary of the trial. Then there's an introductory section, which will kind of explain, be a sort of a preamble explaining sort of background to the disease and the reason why the researchers decided to do the trial. And then there'll be a method section, which is really kind of, well, there are two most important sections, the method section and the results section. So the method section should describe the way the randomization was done. Usually researchers often use computer randomization, but there are other ways like toss of a coin and things like that. But you want to be able to see evidence of how the randomization was done. And also an important thing about the control, I haven't talked about that yet. You want to be sure that the groups are being treated in the same way. So the control group, the control group can be different things. It can either be, most often, if you're trying a new drug, you want to test it against a placebo, and that's the way to find out the sort of full effects of a treatment. And so, say you're using a drug that's in tablet form. So your test group will be given the tablet, which contains the active drug. So your control group also needs to be given a tablet that looks identical to the test tablet, and it should even, if the test tablet is flavoured or has an aroma in any way, then the control tablet should be identical in those respects as well. Because if they're not identical, then that would mean that the blinding was broken, because the people giving the treatment would be aware of whether the dog was getting the placebo or the test drug. So it's important to be able to read about these facts in the trial report. There'll be other things, for example. So I don't know, if your test group was being given the treatment and also maybe it was being exercised regularly or something, it would be important for the placebo group to also be exercised in the same way. Otherwise, there'll be an imbalance in the way the two groups are treated. So that's when a placebo is being used as a control. Sometimes, when there's an effective treatment in existence already, it's helpful for prescribers to know how the new treatment compares with the existing treatment. So, for example, if it's a new non-steroidal anti-inflammatory drug, well, you would want to know how it compared with placebo, ideally, but you'd also want to know how it compared with the existing non-steroidals that you use. As long as that existing one had also been compared with a placebo, so that it had a proven effect, that would be really important to know. So in some trials, the control group is a known effective treatment. But again, it would be really important that nobody's aware of which dog is getting the test treatment and which is getting the control treatment. So both treatments would need to be identical. So if one treatment was, say, the standard treatment, the one we know that's effective is usually given twice a day, and the test treatment is a once-a-day treatment, then you'd need to give the once-a-day group of dogs a second dose of something that looked the same, so that it appeared that both groups are getting exactly the same, if you see what I mean. And you'd expect to see all this described in the methods section, so that you know exactly what's been going on, so that you can trust that, yes, I can see that, the researchers would have been unaware of which dogs were taking which, when they're making the measurements. And it's also important because very often in trials, the dog owners are involved in making measurements. For example, there's in assessing pain, for example, they'll be asked to make measurements that give an indication of their dog's pain level. So it's important that they also are not aware of whether they're giving their dog an active treatment or a control drug or placebo.
Yeah. So Lynn's just asked a really good question. So should we be concerned about studies funded by manufacturers as opposed to university independent research? Not just because they're funding the research, but because they can control what is not published.
Yeah, that's a very good question. I mean, it's there's not an easy answer because people have done studies on this, and they have in human medicines and human published trials and veterinary published controlled trials, and found that yes, sponsored trials are more positive results are more likely to be associated with sponsored trials. Therefore, yeah, studies that are sponsored are more likely to have positive results published. And that could be due to various reasons. It could be that, that companies are doing higher standard trials. I mean, a lot of companies are doing very good trials. In order to get a marketing authorization for a drug, companies have to do randomized controlled trials, and there are standards set by the regulatory authorities. And as I described earlier with the galiperidone drug, they did have to do a sizable trial. Mm-hmm. So sponsorship isn't a definite indication of an unreliable result, because companies do very good trials and required to do in many instances. On the other hand, yes, I mean, a big problem with randomized controlled trials is that not all randomized controlled trials are published, and we don't know what trials aren't published, unfortunately. There are other pieces of research that show that yes, positive results are more likely to be published. And that kind of makes sense, because if you've got a vested interest in producing a product and you're doing trials, then you aren't going to want to show the sort of negative results of the trials. You're only going to want to show the positive results. So if you're seeing a lot of small trials with positive results, then I don't know, that doesn't still mean a lot of confidence. One thing that systematic reviews try to do is to try and sort of find out about the unpublished evidence. but that's a really difficult thing to do. I mean, proper, thorough, systematic reviews involve doing a search of the published literature to find every possible published trial, and also contacting researchers, hand searching, contacting companies, doing as much as possible to try and unearth any unpublished randomized controlled trials as well. But that's obviously a very time-consuming, laborious thing to do. And, you know, there are various techniques that can be done. There's a thing called a forest plot, which when you've gathered together all the results of the trials, I mean, if you've done lots of trials, because I said you've every time you repeat a trial, you get kind of maybe a slightly different set of results, and you'd expect it with small trials. I mean, I said it's going to be difficult to explain, but the true effect of a drug is going to be, say, you know, along that line. And then if you're doing very small trials, every time you repeat a trial with a small number of subjects, the results are going to be sort of scattered quite widely either side of that sort of true result. And as you get bigger and bigger trials, the results should be becoming more and more accurate and closer to the real effect. So what some researchers do when you've got a lot of data, and this happens in human medicine, it's not so easy in veterinary medicine because there aren't so many trials, but researchers have sort of plotted all the results, and in what's called a forest plot, because it's a sort of like a tree going from the sort of small trials up to the, you know, the bigger trials, and when they found, what I haven't explained is that either side of that sort of middle line, you get positive and negative results. So with the small trials, they should be sort of scattered widely from positive to negative. And if what some researchers have found is that, you know, there are missing dots on the negative side, which suggests that, you know, a lot of negative results are not published. And that's a thing called publication bias. And there's not really a lot we can't do much about that. You can only see what there is. Another thing about randomized controlled trials, though, is it is important to find as much as you can. There's no point just picking one trial, looking at the results, especially if it's a small trial, and saying, oh, look, this is a positive result, because you might have just found the sort of outlier, the one that, you know, and that's what we do in Veterinary Prescriber. Every time we review a drug, we do a really thorough literature search to make sure that we've found all the published trials that we can find. But it's a problem, because a lot of research isn't published, and a lot of research that is published isn't very high quality. Oh, and I could mention now, there's been a campaign that started in human medicine called All Trials, a campaign to get all randomized controlled trials registered, so that way, you know, everybody would know what trials were being started, so that if they weren't finished, there'd be people looking, you know, wanting to know why not, if they weren't published. So yeah, it's a trial called the All Trials campaign. There's a website, and I know veterinary medicine, and a lot of sort of researchers and academic institutions, bodies, the RCVS knowledge, and so on, have signed up to all trials to encourage the same in veterinary research. I mean, that would be ideal if all research, all randomized controlled trials were registered with people who intended to undertake them, registered them, and said, this is what we're going to do. Then trials wouldn't disappear, and we'd see the negative ones as well as the positive ones.
Yeah. So slightly moving on from that question, Hannah's asked, when owners are involved, how do researchers minimise the risk of a placebo effect? So getting owner bias from the owners involved in the trial.
Yeah. By including a placebo, I suppose that's the only way, because then you're in the group of animals that have been receiving the placebo. That's when you're accounting for the placebo effect, and you can subtract that from the effect of the active treatment then, and say, sometimes there is a very big placebo effect. You're saying, well, the effect of the drug beyond that is actually very small. Most of the effect we're seeing is placebo effect. And so you have to have owners that are blind to the allocation, and that's how you measure the placebo effect.
I suppose when you're doing a really robust trial, you wouldn't want the results to be based on the owner's opinion anyway. You would want to have an objective monitoring.
Okay. Well, that's another aspect. What do you measure in a trial? That's another very important element of what you're looking for when you're evaluating a trial, and that's something you'll be looking at in the methods. What have the researchers chosen to measure, and why? So in some diseases, it'll be quite simple. For example, I've just been reviewing a new drug for treating mast cell tumours. Yeah. So that's very straightforward, because dog's got a tumour, you give it the treatment, and you're looking to see whether the tumour has completely disappeared or not. And that's a very kind of, it's quite a black and white end point. But in other diseases, and in osteoarthritis, it's very tricky, because there are kind of lots of things you could be measuring. And as I understand it, I'm not an expert in osteoarthritis, that researchers haven't really fully agreed on what are the best measures, what are the most meaningful measures in osteoarthritis. And of course, there are subjective measures and there are objective measures. And the subjective ones are when it's your opinion about whether your dog appears better or not. Whereas an objective one is something like I can't remember the force plate when you're measuring. What's that term? I can't remember.
Putting pressure through different feet. You have the stamp analyzer, or we might have some kind of they have various assessments of lameness and things like that. Arthritis is very difficult. Pain management is very difficult to put in because you can't ask the dog what's your pain out of 1 to 10.
Yeah, exactly. So in osteoarthritis, I know some people say, it's best to have an objective and a subjective measure in there. And also, when you're using measurements, they should be validated measurements. So they should be ones that have been proven to correlate with actual changes in pain. So I understand that the canine brief pain inventory is a validated measure of pain. So that's one, it's a reliable indicator. Because sometimes when you read trials, they're difficult to understand, to be honest, because researchers use all these different measurements. They often don't explain why they use them, or the rationale for using them, and all these different, you don't really know what they mean. You don't know what the changes that they've observed really mean at the end of the day in terms of how your dog is. It really can be quite a difficult area to kind of make sense of. Really.
Yeah. We've had a really good question from Dee. I'm going to make it a bit more of a broad question. I hope you don't mind, Dee. But she's asking very specifically about a certain drug for her dog. But as an owner, where can they find out if they've been prescribed a drug by a vet? Where can they find out about the drug, so the effectiveness of the drug? Or do they just trust in the vets, what the vets are saying about the trials? Because it would be a lot as an owner who has necessarily no scientific background to go and have a look at randomised controlled trials, because they are quite wordy. Even as a vet, they're quite hard to get through. So what would be your advice in that instance?
Well, I mean, your vet is well, is probably likely to be, well, most of the time, like to be prescribing a medicine that's is licensed in this country. And so in some ways, we don't have to worry so much about that because the regulator's doing the job for us to a certain extent. So, as I've said, if it's a licensed drug, then the manufacturers had to prove to the regulator that the drug is effective and safe and is of high quality, and that's that's the reason for regulation, really, so that we can trust that what we're using, the vets can trust that what they're prescribing have been tested and proven to be effective. The difficulty comes then, what we're talking about then is drugs that have been authorized for specific uses, because the authorizations always relate to particular uses. So if we were talking about a non-steroidal anti-inflammatory drug, their licensed uses are usually to reduce pain and inflammation in a dog with osteoarthritis. I think that would be a sort of a general indication, and most of the drugs in that class have a sort of a similar indication. So if it's an authorized drug, you can trust that there is good evidence that it works. That's why it's got an authorization. Sometimes vets have to use unauthorized drugs or drugs that are authorized for other things, for a different use. But there is a lot of information available to vets that so as well, veterinary prescriber, we summarize information that vets can use that can help them to make decisions about using drugs in certain situations. But there are other resources that vets have access to, like the small animal formulary, and that will summarize evidence as well. So and vets will themselves go and look at randomized controlled trial evidence. It doesn't hurt ever for a pet owner, I don't think, to ask why not ask what is the evidence for this. So does that answer the question?
Brilliant. Yes, thank you. I think, from my perspective as a vet, I would say that I wouldn't expect any of my clients to be looking at trials, and that I'd be very happy for them to say, what evidence do you know about with this drug? Also, what's your experience of using it? And what are the known side effects? And ask those sorts of questions and not be afraid to ask, because, at the end of the day, you're giving the vet should be the advocate for the dog. They should be the dog's voice for you and giving you the best treatment that they think is most appropriate for that pet. So I don't think many vets would mind if you asked a few questions about a particular drug. And also, I don't think, I've never been too proud to say, I actually don't know the answer to that question, but I can go and look it up for you, and then I'll get back to you and let you know. I think that's a good idea to ask those questions, but I certainly wouldn't expect any of my clients to be going and having to read trials, because they are hard to read, and with a veterinary background, they can be quite hard to interpret.
It's difficult. It's laborious and time consuming. It's not. You need to know what you're looking for. Pardon?
That's why we like it when you're here, because then you can do a lot of the job for us.
Well, exactly, yes, because it takes time. You need to be knowing what you're looking for. I've been doing it for a long time, but I take advice from other people. I'm not an expert in the various diseases. So, for example, I talk to people who know about osteoarthritis to know about the outcome measures and so on.
Brilliant. So is there anything more that you want us to talk about? Any more other important things that we need to know about randomised?
Well, the other thing, if if anybody ever is interested in looking at a randomised controlled trial, and some people might want to do that. The important thing is, maybe just talk about this briefly. As I said, a published trial is set out in a standard format. It's got the methods, the results, and begins with introduction, ends with a discussion. And the important thing, I think, is not to kind of just go and read the trial. I mean, often I mean, in some ways, published randomized controlled trials can be used as sort of a bit of a promotional tool. They're there to promote the ideas of the researcher, and some researchers might have interpreted the result in a very sort of optimistic way. So if you're looking at a randomized controlled trial, you always want to think, what do I want to know? Not what has the person found that they want to tell me about. What do I want to know, and can I find the answer in the trial? So that's the thing to do, not to be led too much by what the report writer is telling you should take away from it, because that can be misleading and not give you the answer you want. Yeah.
I think I've learned a lot. Hopefully everyone has. I know you had a few slides that you wanted to go through for just a checklist of resources, didn't you? Shall I pop those slides up?
Yeah, okay, yeah. Oh yeah, I just put this is just for any, I mean, when you're looking at a randomized controlled trial, it is kind of, it can be difficult to remember what you're supposed to be looking for. And, several organizations have published sort of checklists of the, sort of crib notes for the things that you should be looking out for. So I've just listed 3 of those there. There's, I think it's a very good one, published by the Centre for Evidence-Based Veterinary Medicine in Nottingham University. There's an organisation called CASP, the Critical Appraisal Skills Programme. That's really a sort of evaluating human medicine trials, not specifically veterinary ones, but it's the same principles. And there's RCVS Knowledge. They've got sort of tools and checklists on their site as well. So that's just for anybody who wants to have a closer look at randomized controlled trials and have a go at evaluating them. Oh, do I need to move to the next one? Yeah, I did need to do that. Yeah. How do I do that? Oh, here we are. Look, I've got to go to my, that's it. Oh, that's just the, I mentioned the All Trials campaign. You can go on there and support the cause. This was just something to show where randomized controlled trials sit in the sort of evidence hierarchy. So here we've got evidence going from least reliable to most reliable. So at the bottom, we've got anecdote and opinion. We talked at the beginning about why that can be unreliable. We shouldn't rely on it. Above that are types of studies called observational studies or case reports. They can be useful for certain things, but not finding out whether a treatment works. And that's where randomized controlled trials sit. And then above there are meta-analysis, which is when you've done a systematic review to find all the randomized controlled trial evidence on whatever it is you're interested in. As long as all the trials are reasonably similar, you can do a sort of a statistical analysis where you combine all the results together to get a more accurate estimate of the effects of a treatment. And that's the sort of most reliable type of evidence. And then the last one, these are just the websites of, the veterinary prescriber website, which is aimed at vets mainly, and we've got another website called Veterinary Medicine CPD, which is aimed at veterinary nurses, SQPs and pharmacists. And these are independent resources, and if you don't mind me saying, having an independent resource is really important because there's a need for somebody to go and scrutinise evidence and find out the flaws and the biases and bring it to people's attention. So
absolutely,
you need to use veterinary prescriber.
Yeah, it's an absolutely brilliant resource. So for any vets, nurses, etc. out there who are wanting to know a bit more about certain medications, then vet prescriber is invaluable, I think. And as you say, it's independent. So you're not being swayed by the company who's trying to sell the drug. You're getting the best evidence, and in a really easy to read way as well. So
yeah.
I'm on a lunch break, I can quickly flick through and read about something. I'm not trying to delve into a study where you need to sit down and I have to have the study printed out, a highlighter, and really it takes a lot of time to go through each study. So you've done all that hard work for us, which is
yes, intended to save time. Yeah, exactly.
Thank you so much, Andrea, for joining us and for a really informative, brilliant talk on randomised controlled trials. If anyone's watching this and has really enjoyed it and wants to know a bit more, then do look back at our previous live we did on evidence-based medicine, because that was also brilliant. Andrea's really engaging and lovely speaker, and so knowledgeable. So we're very lucky to have her as a resource here at CAM. And thank you for joining us here tonight.
Well, thank you very much. Thanks for inviting me. Good to be here.
Right, everybody.
Okay. Bye-bye now.